Yeah. Okay, somebody else will take tapes then. So it's can't call it phone. I can't really call it tape. Recording, yes.
Yeah.
I still have 3 of them. I may pull the fiberglass one out for the season. The plastic one is long. And then the sit on top is too cold for sit on top. Pull that. Well, I sure hope this, because we told them more than a month ago. I mean, at the 2-week mark, just still haven't told them.
They'll have it done soon. I think you like to do it sometimes. It's usually the summer to get it done.
So as soon as possible. You know, because one was going down. So 2 weeks later, it's still in water.
Oh, this is weird.
Yeah, you know, last year, kind of low water, but all season. It was forced throughout until the very end. And we're lower than the entire last year.
Yeah. Go ahead.
Yeah. No, not the YouTube video. It is. I screwed up, but it is open now.
It's open now.
I'll tell you what I did later.
We have wait a few. What do you need to do?
What happens is, you know, holidays.
All right.
Children, kids will come over.
I'm gonna go ahead and get started here. My name is Thornton Ritz. I'm a PhD student at SUNY ESF. I've done now three summers worth of research here on the Thousand Islands. My advisor, John Farrell, is here today. He's a big part of the research that I've done and the research that goes on at TIBS. And he's graciously allowed me to share some of the big picture work that TIBS does, as well as some of the new work that I've done for my dissertation. I would characterize myself as an aquatic ecologist that specializes in early life stages. So we're going to be talking a lot about baby fish today. So hopefully everybody is excited and looking forward to hearing more. So before I get into wetlands and some of the work I do, I just wanted to highlight the Thousand Islands Biological Station. If you don't know, TIBS, right across the channel from us, located on Governor's Island, is a field station run through SUNY ESF. And we focus on conserving aquatic resources using ecosystem-based science. We work with decision makers, help inform decision makers, and provide really exceptional education opportunities for someone like me, graduate students, undergrads, high school students, elementary school students. TIBS is a really, really great place for science in the Thousand Islands. And like that sort of spiel says, we really focus on this collaborative research approach, right? So we work with agencies. We work with the New York DEC, the U.S. Fish and Wildlife. We work with amazing universities, SUNY ESF being the prominent one, but we've done work with other SUNY schools, Clarkson University, Carleton University in Canada. We have a lot of collaborations all around. In terms of local groups, we work with the Thousand Islands Land Trust, Antique Boat Museum, and Save the River. So we have a lot of different partners here on the river. We also have a new program here at TIBS called TIBS Friends Program, and this is sort of supporting the future of TIBS and the future of research in the Thousand Islands region. And then I put in the public here, and that's everybody here who's coming to see talks, collaborate with us, maybe give us access on private lands, come see the station, things like that. All that, may not seem like you're contributing to the direct science, but just learning about these topics and being present and understanding what's going on is really important to us at TIBS. So you guys are part of this as well. So where do we conduct fish research in the Thousand Islands? And I want to preface it and say fish research. At TIBS, we do all kinds of research that I could do a whole talk on. We do everything from fish to aquatic plants to muskrats to water quality. But since I'm a fish guy, I have a kind of a bias towards fish. We're going to focus mostly on fish today. So in general, what sort of surveys do we conduct on the river? So early spring, we're spending our time in the embayments of the main river. This is where we're trying to collect muskellunge, part of our muskie recovery program, in terms of getting gametes to fertilize and spawn and stock muskie. That's a big initiative we have. In addition to catching muskie, we catch the whole adult fish community, largemouth bass, smallmouth bass, different types of catfishes, all these species that we get data on. And that's really important to understand long-term changes in the river. After we're done with these embayment surveys, we move to the wetlands. And the wetlands are this
I don't know what an embayment is.
Sure, so a bay, so like Flynn Bay, French Bay Marina, so a bay on the river. Then moving to the wetlands, this is what we consider nursery habitat, and you're going to hear a lot about that today because that's the focus of this talk. But this is where we look at the young of year fish community, and what I mean by the young of year fish community is the fish born that year. So if we're sampling these wetlands in June, those fish were born in May or April or June, okay? And in the wetlands, we focus on a number of native species, but our focal species, the northern pike, is a prominent feature to our wetland surveys. And we'll talk about northern pike later in this talk. And then finally, after June, after we get through June and some of July, we then move to these littoral zones or your near shore area. So think all around all the docks, the thick vegetation in the shallow water. You'll see us out there with the seine net trudging through the silt and sediment and aquatic vegetation in wetsuits, and we're there surveying the young of year fish community that's present in these bays. So we work in a number of different habitats, from adult fish to young fish, throughout the growing season. And today we're going to focus in on our wetlands, our nursery habitat, and fish emigration in the Thousand Islands. So first off, a little bit about coastal wetlands. I'm going to call them coastal wetlands. A lot of people, when they say coastal wetlands, they think the ocean, but we have a number of coastal wetlands here in the river and in the Great Lakes. And when we think of wetlands, we think of these three key characterizations of them. So we need the presence of water during that growing season, right? We need water during the springtime when plants are growing, when organisms are hatching, when you have all this biological activity. You need hydric soil, and what this means is you need soil that is saturated at some point during that year. So it really connects to this presence of water. That soil saturation allows anaerobic or processes without oxygen to occur in the soil. This type of soil and processes that go on in the soil really facilitate aquatic vegetation. So aquatic vegetation is our third key piece to wetlands. So if you go look at any of the wetlands around here, French Creek, for example, right in town here, you can see the aquatic vegetation in French Creek. And we'll learn that all of this is kind of key to characterizing and having a healthy wetland ecosystem. And when we think about these wetlands, we think about environmental conditions. And I'm going to talk about these environmental conditions a lot today. We think about things like water temperature, dissolved oxygen, or the amount of oxygen in the water. Water level is, I know, a topic that a lot of people care about around the St. Lawrence River and is really important. That has its piece in all of this. And many of these environmental conditions are driven by how the wetland was formed. So some of these wetlands will have formed during glaciation, causing certain conditions. Some of them are still forming today based off of environmental conditions. And I'll show you a few examples of different types of wetlands that we have here in my next slide. But these environmental conditions are dictated by how the wetland was formed, what it looks like, where it is in the river, where it is compared to a bay in the river. All of this stuff is really important for characterizing the conditions in that wetland. And then obviously wetlands have very high productivity. There's a lot of nutrients. This has a ripple effect to the whole entire food web. We have lots of aquatic insects, zooplankton, fish, as well as a number of biota that I'll talk about here today. These are just some of our beautiful wetlands here on the St. Lawrence. You may be familiar with them. We have Cranberry Creek. We have Flynn Bay, which has a beautiful natural wetland. This is just a section of French Creek here in town. All of these wetlands were formed in different ways, and that sort of characterizes what goes on, the conditions that are in these wetlands. So more specifically, we define these wetlands as hydrogeomorphic types. I don't want to get sort of lost in that, but essentially these wetlands interface with, for our example, they interface with the river, and you have different water flow regimes that affect the wetlands. So for example, we can have an open embayment wetland. So this is something like Eel Bay, a little bit downstream from here. You have a large wetland that allows winds and currents to build, and that water pushes in and out of those wetlands. You could have a barrier beach wetland, so you can have sand that accumulates near a wetland area and turns that area into a shallow, seasonally wet area. You can have drowned river mouths. Drowned river mouths have more of a glaciation type formation. So during glaciation, glaciers melted, and a lot of these drowned river mouths were flooded and inundated with water. And so those river mouths, the water went up and the surrounding area became really wet and seasonally wet, like we see. These drowned river mouths are very, very productive. They have a lot of nutrients, a lot of different organisms that call these places home. They're all very productive, but especially these drowned river mouths, which we have a number of drowned river mouths around here. And we'll talk about those later in this talk. And we can have protected embayment. So you can see here, this little island can completely change the environmental conditions that are present in that wetland. That little island could block wind, current, and this is going to influence the water exchange that is going on in these wetlands. So again, these conditions dictate what's in these wetlands. So when we think about wetlands, why do we care about them? Why are they important? We like to think about what we call ecosystem services. And when I think about ecosystem services, I think of sort of a process or an interaction that has a positive benefit for that ecosystem. And so these can be a number of different, services that wetlands provide here. And remember, these wetlands are present throughout the Great Lakes. So you can see, I know it's a little small, but we can see lots of orange dots in our Thousand Islands region. But these wetlands are spread throughout our Great Lakes. And really, really provide a number of amazing ecosystem services. So the number one that we really think about is water quality, right? So these wetlands act as a filter. They can hold, store nutrients to keep excess nutrients kind of at bay, so they're not going, say, into an embayment. Through biogeochemical processes, we can process these nutrients so we don't have excess, too many nutrients coming into an embayment. In addition, we can store groundwater and groundwater recharge. So if we have a really low water year, some of these wetlands can hold water that can act to replenish low water areas. In addition, wetlands provide protection, and I think we've all heard about the impact of mangroves, wetlands on the coast. We have similar effects here in the St. Lawrence River and in the Great Lakes. We don't have mangroves, but we have wetlands with aquatic plants and bank stabilization that buffer the shoreline against waves and floods. Wetlands are a key piece to protecting our coastline and erosions, not only in a marine system, but right here in the Great Lakes, right here on the St. Lawrence River. In addition, I sort of spoke on this, but wetlands are highly productive, and this translates to the entire food web. We have fish nursery habitat, which is the topic I really like to talk about, and you'll hear more about that as we work through the talk here. But these wetlands are also home to waterfowl, shorebirds. We have we have waterfowl migrating into these spots right now. We have shorebirds laying nests, resting, feeding in all of these wetlands. I can't tell you the number of times I've seen American bitterns and all kinds of amazing wetland creatures as I've been out in these spots. Reptiles, amphibians, you have something like the Blanding's turtle here, which is, endangered and really uses these wetlands as its critical habitat. And then we can't forget our aquatic and terrestrial plants. I know plants maybe some people really love plants, and plants aren't the most interesting thing, but these plants are critical to everything that goes on in these wetlands. And they actually influence the environmental conditions pretty distinctly, and you'll see how that happens. And then thinking about the human dimension, right? We have these biocultural uses. So there's a big push in the Great Lakes to develop wild rice in coastal wetlands. These wetlands have human values as well, cultural as well as recreation. So thinking about things like fishing, paddling out in these wetlands. I've spent a number of hours kayaking through these wetlands, and it's just remarkable, and it sets your mind at ease. So there's a human dimension to these ecosystem services as well. Not to just say that it's all about biology and things like that. There's also a human dimension that makes these wetlands important. So I keep saying this word, environmental conditions. What does this mean? So these are the conditions that dictate a lot of the processes and what sort of organisms, specifically fish, in the context of this talk, that can live in these wetlands. So the first sort of environmental condition we think about is water temperature. So if you went to one of the wetlands in June and jumped in and went swimming, it would feel pretty good. If you jumped in the main channel in June, I think it would be pretty darn cold. So these wetlands, they're warm. They're warm and shallow, and this really facilitates fish and the prey that fish eat, their survival and growth. So water temperature is something we pay attention to. Water level. So obviously there's water level regulation on the St. Lawrence River, and I know that's a significant topic of conversation in the area and throughout the Great Lakes. And TIBS and other organizations, other scientist groups, things like that, have found a significant influence of water level and water level regulation on the St. Lawrence River. We've seen shifting plant communities. We've seen a disruption in these natural hydrologic cycles. And I want to sort of dig a little deeper. And so what else interfaces with water level and water temperature is in this sort of context as well. But I want to dig a little deeper past water level to think about some of the research that I do and some of this new in a sense, sampling that we're doing here at TIBS for dissolved oxygen. So what is dissolved oxygen? Dissolved oxygen is the amount of oxygen dissolved in the water. Pretty simple as the name states. So this can be expressed in milligrams per liter, or what I think is a little more easy to understand, a percent saturation. So that's the percent of oxygen that is in that water, okay? And some of the key pieces to this and a key relationship that we have is that warmer water holds less oxygen, okay? So as the water warms up, you have molecules moving around really, really fast in that water, creating energy. And then that energy allows that oxygen to be able to break the barrier that the water is holding it in and go to the surface and just, go out into the air. So warmer water holds less oxygen. I like to think this analogy, you're in a room full of packed people, everybody's moving really slow. It's going to be a lot harder for you to get out of that room. If we're in this room and we're moving around like crazy and you can move really fast, you can get out of this room really quickly, okay? So the key thing is, as that water warms, and especially in these wetlands, we lose oxygen in the water, okay? So something to think about as we move forward with climate change, things like that. And there's this buzzword that's involved with dissolved oxygen, and I think it, you guys may have heard of it before, but it's this term hypoxia. So hypoxia is less than 3 milligrams per liter of oxygen, or approximately 30% oxygen. So hypoxia is this term used to characterize low oxygen. So below this level, most aquatic organisms run into problems. They get stressed out, they can't handle these low levels. And we see the deoxygenation of the ocean and hypoxia in the Gulf of Mexico are these big headlines. And I want to emphasize that some of this is also occurring in the Great Lakes and in the St. Lawrence River. We're still working on figuring out exactly the causes and the mechanisms and the impact, but this is occurring, and I think will continue to occur during the onset of climate change. And so why is oxygen important for fish? So just like fish, just like humans, that fish respire and they need oxygen. This runs their daily metabolic processes, just being able to live, to be able to escape predators, to be able to get prey. Oxygen is a key piece to the metabolism of a fish and a fish's daily life. So now we have a little sense of what characterizes our wetlands. So what sort of processes do we see in these wetlands? And I hope this is sort of new information to everybody, so when you drive over these wetlands, you're going to be like, oh, I see that these processes can happen, and wow, it's more than just this swamp or this really nasty, muddy wetland. So let's take ourselves to Cranberry Creek right here, okay? We have the bay right here with some nice big adult fish. So say we're in April and May. During this time, the water level goes up, right? We have springtime melt, we have weather patterns in the spring, ice melt, so water level increases. This causes our adult fish to move from the main river, go into our wetlands, spawn. They lay their eggs. The water level eventually starts to recede as we move into the summertime. So as we move in past April and May, the water level recedes, causing those adult fish to say, I got to get out of here. I got to go back to the main river where I can be safe and I can live. So those fish eventually go do their thing in the main river. So come June and July, what is going on in these wetlands? We have lots of little baby fish now. They've hatched, they've gotten through the egg phase. They're tiny little 1/4 of an inch fish. During this time, our water level is still decreasing. We also see decreases in oxygen, like I just sort of spoke on, this idea of dissolved oxygen and the amount of oxygen in the water. And coinciding with that, we see increases in water temperature, okay? So these fish are now facing higher water temperatures, lower oxygen levels. So what does this cause them to do? It causes some of them to leave and move to the main river and live in the main river for the rest of their lives, okay? So species like northern pike, our largemouth bass, a lot of them are going to be born in these wetlands and eventually leave the wetlands and call the main river the bays. Everywhere you see out in the main river, they're home. Some of the fish don't make it. That's just a natural process. Some of these fish aren't going to make it out of these wetlands, whether they get eaten, whether they don't have enough food, perhaps the environmental conditions didn't favor them. Some aren't going to make it. But I want to emphasize that there are going to be some fish that can live in these wetlands year-round, okay? They're going to be tolerant of high water temperature and low oxygen. A great example of that is a grass pickerel. A grass pickerel, similar to a pike, but it can tolerate a lot lower oxygen and a lot higher water temperature. So Cranberry Creek, full of grass pickerel, will grow to about 6 inches, but they call this wetland their home for the year-round. But I do want to emphasize that the bulk majority of these fish emigrate out to the river. And this connection to the river is key, because without these baby fish, young of year fish, moving into the main river, we would not have as many adults. and as many adult populations. I don't want to say that all the baby fish come from these wetlands, but a huge portion of the fish that eventually reach adulthood that people are catching on their boats and having such a great time come from being babies in these little wetlands. So why do we call the wetlands fish nursery habitat? So I've sort of touched on this, but we have really advantageous environmental conditions. We have really, really warm water. This warm water is going to facilitate the growth of prey. It's going to facilitate the growth of those predators. In addition, we have protection and shelter. So some of these wetlands are tucked back in away from the main river, and we can get protection from currents, from wind, from human disturbance. Okay, imagine a little baby fish this big in the middle of the channel as one of those ships goes by. It's probably not going to be the best place for it, but tucked back in a wetland in Cranberry Creek, it's going to be perfect. So they have lots of different food resources in the wetland. Probably all of these fish, when they're little, they eat little zooplankton. So think of your big pike that you see gobbling down fish on YouTube. That fish started this big, eating zooplankton in the wetlands. So these wetlands really help facilitate zooplankton, macroinvertebrates, and other fish species that are used as prey and forage for these baby fish as they grow and develop in the wetlands. And I want to note predation happens. You know, these wetlands are a dangerous environment for a fish, but compared to the main river, it's a much safer environment. And also, you know, predation is sort of a natural part of the food web and the processes that go on. So that aspect of fish being able to eat little baby fish in these wetlands is just sort of a natural cycle. So they just provide a really nice environment for these fish to live in. And as I said, it provides the foundation for the main river fish community, okay? So these wetlands are critical for the future of adult populations on the river. So what is the current state of the wetlands? So in general, in the Great Lakes, we have about 50% loss of coastal wetlands. A lot of this is attributed to development. You know, we have fragmentation, breaking up wetlands. We have just building of impervious surfaces like pavement where water can't drain as well. We have draining for farming, and all of this development, sadly, wetlands have been exposed to it. In addition, and really in the Great Lakes, and it's very evident on the St. Lawrence River, we have invasive species. So we have an invasive hybrid cattail here. And this hybrid cattail, pictured right here, is really detrimental to the wetlands. They're very thick stems, they're very dense stems. You have a high number of stems coming from one plant. This clogs the wetlands. It makes it physically impossible sometimes for adult fish to move in and spawn. It also influences the mixing of the wetland. So less water is going to be able to move into that wetland and maybe recharge that wetland with fresh oxygen or cooler water. So typha and these cattails that we see, I'm sure people have seen floating mats of these things on the river. They can have a significant influence on the young of year fish and fish in general in these wetlands. And then obviously water level, we all know water level has these negative impacts through this disruption of natural hydrologic cycles and processes on the river. And again, water level interfaces with these environmental conditions like DO that I'm talking about. And then, you know, thinking about the present and the future climate change, right? So we have this really tight relationship between oxygen and water temperature. So if we see continued increases in water temperature, so this is some TIBS data. The x-axis, your horizontal axis, starts in 2004 all the way to 2021. And we've seen about a 2-degree Fahrenheit change. And this is from the main river. But this definitely translates to the wetlands. And the wetlands are even shallower. They can warm up faster. So this problem could even be happening more intensely in the wetlands. And then obviously different weather patterns. You know, this summer and the last 2 summers I felt on the river were characterized by, you know, low rainfall, things like that. And we can see the impacts. You can see some sediment coming up here in the wetland. So all of these wetlands seem to be impacted in one way or another. So at TIBS, how do we combat this? So we use we have this program and we have this sort of conservation strategy, the fish habitat conservation strategy, and it allows us to really develop and plan and then go out and implement restorations in the wetlands. And once we implement the restorations, we then monitor these conditions to make sure that what we're doing is actually helping the wetland. So how do we do this? So we actually, not me, but the U.S. Fish and Wildlife, some of our collaborators, we can access these wetlands through organizations like TILT and bring in excavators. And we actually dig out channels in the wetlands. And I know this may seem really, really destructive and horrible to have this big excavator in the wetland, but we are removing so much of this invasive typha that is clogging channels. I'll show you a photo here next of what this actually looks like after we've done the work we do. Another key thing is exposing these banks, exposing this sediment actually exposes the seed bank. So this allows a lot of native plants to come back and to combat these invasive typha. So yes, it looks kind of destructive, but we have really great results. So if you take a look on our left here, this is pre-project. You have two different panels up and down. So if you look at our first one, we can see lots of cattails, even in both of them. You can see solid, right? It's solid, that yellow color, that's going to be cattails. After we bring these excavators in, we can carve new channels. So now we have water in places where there was cattails. These cattails are usually on mats, so there is actually water underneath the cattail, but it's very sheltered by the typha itself. And we speculate that fish move underneath it, but it's definitely nowhere in comparison to the benefit of open water. So we modify channels, we create connectivity. We also create what we call spawning pools. So you can see within this solid piece of typha mats, typha wetland, we can go in and we can create these little pools, okay? And these range anywhere from 2 to 4 feet deep, something like that, depending on the water level, and they provide great spawning locations for fish, and then ultimately a little nursery habitat for our baby fish, okay? So these projects create a lot more habitat in locations that were once clogged with cattails. Here's another really amazing before and after. So this is Point Vivian. So you can see on the left, we have this tiny little channel, and the cattails have kind of invaded everywhere else. And then post-project, look at all this water now. So we have a lot more access for adult fish and then baby fish. And I want to note, this is really cool. So you can actually go to Google Earth now. You can do it on your home computer, and you can go back in time and see some of these sites before we went in there and did the restoration. So if you look along Cranberry Creek, French Creek, Chippewa Creek, Point Vivian, you can see amazing changes like this, where we've gone from all cattails to water and to fish ultimately, and other wetland species. So we do all these restorations, we create all these new habitat in the wetlands. What do we do next? So this is where the research and the monitoring really ramps up. We need to monitor these environmental conditions and what's going on in the wetlands. And what I mean by that is we go out and we collect data, right? We're going out, we're measuring water temperature, we're measuring oxygen, we're trying to understand the impact of the restorations on these environmental conditions. We do it with fish as well. We survey the fish. I'll get into fish here in a little bit. So I want to know, I'm really in wetlands, okay? We're going to have low oxygen in wetlands. That's just a natural process. But is there too low? Is there a tipping point? If water temperatures change, is the oxygen going to get depleted too quick? Okay, so think of it as this sort of delicate balance between warm water and oxygen. And could climate change impact it? Find out. And then ultimately, like I've said, do our restorations impact these conditions? So when we say reference, it's going to be an unmanipulated channel. It's a channel that is naturally there. When we say modified, that means we've gone in there and directly changed the habitat. So how do we monitor these environmental conditions? So I'm fortunate enough to be coming to TIBS, where we have over 10 years plus, closer to 20 years of some of this data, of instantaneous measurements. So we actually go out in the wetlands, we hop in our canoes, and you can put that probe right in the water and get measurements of the water conditions. the water temperature, the dissolved oxygen. And so this has really allowed us to build a huge foundation of data that has informed many of our restorations. And then more recently, and it has to do a lot with my work, we're using data loggers. So what a data logger does is it allows us to leave these little data loggers out in the wetland, and you can set how often you want it to measure. So I leave these out and they measure once an hour. So I get a water temperature and an amount of oxygen in the water every single hour. And we're going to learn that this is really, really important to understand what is happening in these wetlands. And again, these are situated in kind of key habitat types, right? We want to know if our restorations equate to a natural channel that's been there for years and years and years. So we have to strategically place these in locations to understand the effect of our modifications and also just to monitor what the heck is going on in these sites. So I'm going to show some data here. I'm going to walk through it. I don't want anyone to be confused about what they're seeing, so I'll try to really explain. So let's think about how oxygen changes on a daily basis, okay? So if you look at our x-axis here, we have from 12 a.m. all the way to 11 p.m., so the whole day, and we can see the same sort of squiggly pattern in all those lines. Just focus on that shape first. What is that telling us? So that's showing us that DO rises and drops on a daily basis, okay? Oxygen levels change on a daily basis in these wetlands. So around 9 a.m., we get lots of photosynthesis, lots of plants doing photosynthesis, creating oxygen. So around 9 a.m., we see these peaks of oxygen, okay? Our y-axis here is our average hourly oxygen measurement. So during the year at 7 a.m., this measurement is what we have for oxygen, okay? So we can see that throughout the day, oxygen increases, likely tied to photosynthesis, plants producing oxygen, and then we have this decrease at night, so it gets dark. We lose oxygen in the wetlands. So imagine you hiking a mountain up and down every single day, having different levels of oxygen. That is what is happening in these specific sites. These loggers are stationary, they do not move, so these measurements are all at one individual location. So we have a general pattern related to photosynthesis. What do these colors mean? So these colors indicate where the logger is placed in the wetland, okay? So that blue line is going to be within the bay itself. So you can see, for example, if we look at this left panel here, really high, great, amazing oxygen in the bay of Eel Bay. Moving to our green and our gray colored lines, these indicate locations within a wetland. So our green line is more of an exterior edge of the wetland, so closer to the bay itself. And our gray line is interior, so very far back in the wetland. So we can see in our panel at the left that both the exterior edge, so closer to the bay, and the interior of the wetland have very similar conditions, right? And you wouldn't think that would be the case. So why is this happening? So this is Eel Bay. So Eel Bay is this huge embayment that gets wind and current from the river. So what that is doing, it's mixing the wetland. So we see similar oxygen patterns. And if I put temperature up here, you can see a very similar relationship. So we see, for example, in that left panel, Eel Bay has mixing of water throughout the wetland. Let's move to Flynn Bay. So Flynn Bay is a little different than Eel Bay. It's protected. It's protected from some of those winds and currents. So we can still see the bay itself, very highly oxygenated. But now we see a gradient. So towards the edge of the wetland, we see more oxygen. And as you move further into the wetland, we see very low oxygen. So again, this points to the fact that the location of the wetland matters. How the wetland is orientated on the river, to the wind, to the current, exposure, things like that, dictate these environmental conditions. And this just sort of brings to the point that we have to monitor multiple wetlands. We can't just go to one wetland and say it's the same throughout the river. These different types of wetlands change these environmental conditions. Let's look at this on a seasonal scale, right? So we have this, the seasons here, spring, summer, fall. What do we see? Basic pattern, highly oxygenated in the spring, the time when those adult fish, those baby fish are doing their thing. Moving to the summertime, we see decreases in both locations related to warming water temperature, things like that. These seasonal patterns are really important to monitor too, because again, we can take this data and we can make predictions from it, and we can also understand changes, long-term changes. And again, we can see some separation in our right panel between our green and our gray, while our left panel is very similar. So on a daily scale and the seasonal scale, Eel Bay, exposed to currents and wind, is mixing throughout the wetland. Flynn Bay, it's not doing that. I'm not saying that's bad. It's just a different type of wetland with different conditions, okay? So it's really important to monitor multiple locations, multiple types of wetlands. So what did we just learn? I know I talked through a lot of data, but some basics here. Not all wetlands are built the same, okay? These wetlands and how they formed and where they're located on the river are gonna dictate a lot of the environmental conditions, which is ultimately gonna dictate fish, plants, other organisms in these wetlands. If we think about oxygen on a daily scale, we have a peak during the daylight hours, and at night we have a drastic decrease. So think about a fish living in these places where you're exposed to really high oxygen and then you're exposed to really low oxygen. What does that do to a fish? We'll talk about that here soon. And then seasonal change. We have high oxygen in the early spring, moving to the fall time and late summer with lower oxygen. In the fall, as the water temperature gets colder, we do see a slight rise in the oxygen. So regardless, very important to monitor multiple sites, things like that. So then what about the fish? Time to talk about the best part of all this. So the fish community, how do we assess the fish community? How does TIBS, how does my research assess the fish community? So the fish community is made of those young of year fish, right? We're surveying in June, that fish was born in June or May of that year. We also get resident fish. So how do we do this? We use these little mini hoop nets you can see here. So we block the wetland channels so we can catch fish that are leaving their nursery habitat, right? I showed you that picture of fish moving in and out of Cranberry Creek. We're trying to capture these baby fish as they move out, okay? Fish are identified, counted, measured, some are measured, and then we release them on the other side of the net to continue their life and to continue the process of moving to the main river. We take instantaneous, so we put a probe in the water, we take water temperature, we take oxygen measurements, and again, where these locations are strategic. We want to know if our habitat modifications are helping or hurting the fish. This is about 2,000 little baby largemouth bass, one net, so we can see some real interesting species. And what are they? So we have a number of tolerant, what we would call tolerant wetland species. So these are species that can cope with low oxygen and higher water temperatures in comparison to others. So some of these fish you may be familiar with, for example, the brown bullhead can breathe through its skin or can respirate through its skin. So that's how it combats low oxygen. Our bowfin, a phenomenal native fish species. Maybe you've heard it called a dogfish before, but that's what a bowfin is. These fish can actually gulp air from the surface to live and to cope in these conditions. Similar to a long-nosed gar, our gar can do that as well. And then one non-native in this whole bunch. All these fish are native except this common carp. Carp sadly have become a prominent feature to the river. These are more in our bay wetlands compared to something like French Creek, but they are present. So all of these fish have their own role in the wetland ecosystem. Here are some sensitive wetland fish. So we think about our Cyprinidae. So that's a family of fish. We consider these forage fish. These are fish that pike, other predators eat. We have Lopomus, things like bluegill, smallmouth bass, northern pike. We consider all these fish sensitive. So these fish may not be able to stay in the wetland as long, may die in the wetland if the conditions aren't right. So it's really important to know what you're catching and to understand what you're catching. And I think as you can see, all these fish, you can see the background of my hand or somebody else's, they're all very tiny. So that's, I think, the best part is you get to see so many fish and so many different species because they're tiny and there's so many of them. Versus out on the main river where we're tracking down one adult at a time. So yeah, I sort of like my role with little fish. So what does this look like? What does a fish emigration look like? So take a look at our graph here. Our horizontal axis is dates all the way from early June to the end of July. Early June to the end of July. Our y-axis, our horizontal, is going to be fish catch, right? So what do we see? We see peaks and rises in catch. We generally see a downward trend. So earlier in June, we catch a lot of fish, up to 2,000 per day in one net, or 4 nets, I should say. And then we see a slow decrease to really, really small numbers, below 50. So this gives us sort of evidence of this emigration process. We see a huge bulk of baby fish, young of year fish, moving to the wetlands, or moving out of the wetlands. 26,000 fish. So this was data from my first summer in 2021, and we caught a lot of fish. You could see Eel Bay, definitely a lot more than Flynn Bay. This likely could be related to those environmental conditions, the similar mixing and the similar DO levels throughout the wetland. This could be influencing some of these catch numbers. But the point is, we see peaks and rises based on the species of fish. They all develop differently. They all move into the wetland at different times. How do we understand fish emigration? So there are a number of tools we can use, and I'm going to treat this slide as a little preview of what's to come. This is some of the current data I'm really working on, so talk to me in maybe next summer when I'm up here and I can tell you all about this. But I just wanted to emphasize, we use a number of tools to understand what's going on with these fish. We can look at their size, so we can get a sense for growth. We can look at the phenology, the timing. When do they emigrate? Why are they emigrating then? Why is this species leaving the wetland before another species? And then we can use biomarkers. So biomarker, think of a structure that will tell us something about the past of a fish or another organism. So in fish, we use what's called an otolith. So I don't know if anybody knows what an otolith is, but an otolith is a calcium structure in a fish's head. It helps with orientation and hearing. It's literally it looks like a little piece of calcium. And otoliths, as the fish grow, otoliths grow. So you can see this is an example of an otolith. So you see all these lines like a tree, right? So you can actually age fish. And these lines represent a day. So you can actually take these out of young fish and you can estimate how many days old they are. So we use tools like this. I want to make the emphasis, you probably can barely even see them, but they're small. I'm working with 3-inch long fish. These are like grain of sand in their head. But they tell an amazing story. So we can get at things like age and growth. We can even do more high-tech analysis, and we can actually know. So as the fish grows, it's absorbing what elements are in the environment into this otolith. So thinking about like a carbon getting into this otolith or something like that. So we can actually learn what elements relate to something like hypoxia. So during low oxygen, there's a high amount of manganese. It's an element that's naturally occurring in the wetlands. We can shoot a laser in this otolith and we can see what the concentrations of that element is as the fish grows. So that's what I mean by a biomarker. It can tell us about an event, like a low oxygen event. And also we can identify stocked fish. So at little, small, larval baby fish stages, we can actually, and we do this in our lab, we can mark the fish. So we can immerse them in a bath and we can put a highlighter mark on this otolith and say, that's our fish, we stocked that fish. If it doesn't have a mark, it's a wild fish. So what did we learn about the fish community here? We have this fish immigration process and we have evidence for it. Early June, we see high numbers moving into the end of immigration. In late July, low numbers. Likely related to environmental conditions, location, things like that. And we realize that it's really important to focus on what species they are. They all hatch, they grow, and they leave the wetland at different times. So understanding their tolerance and their life history is really important. And I'm just going to finish up here looking at a species that a lot of people are familiar with, our focal species here, one of our wetland species on the river, the northern pike. So the northern pike are a wetland in the context of the river here, are really this apex predator. in the wetlands, and they have a circumpolar distribution, which means they're found all around the globe. Pretty remarkable fish. I'm sure everybody's used to seeing these this big. These are about this big. So these are, again, our baby pike. And like I said, born in the wetlands, they grow, they develop, eating zooplankton all the way to baby fish, and then eventually move out into the main river. So these fish directly connect to the adult fish we catch in the main river channel and the main river bays. So what are we seeing for northern pike young a year in the Thousand Islands region? So if you take a look at our axis here, our horizontal axis, 1985 all the way to about 2018. We can even update this graph further and we'd see the same pattern. Our y-axis here is essentially fish catch, northern pike catch of young a year baby northern pike. We can see a decrease, right? Is this related to changing environmental conditions? Likely. Is this related to invasive species? Likely changing water levels, things like that. We're really trying to tease out the exact mechanisms that are causing this decline. And briefly, how do we address these changes? So when we think about protecting, we have wetland restorations, right? I talked about excavating channels, spawning pools, and we monitor the wetlands. So looking at abiotic environmental conditions and fish catch to understand what is causing the fish to leave. What is causing that day where we catch 2,000 fish versus a day where we catch 0? Okay. And then, yeah, thinking about stocking. So this past April, we stocked about 150,000 northern pike into the wetlands at about this big. So I'm still working on trying to figure out how many of them survive. So again, thinking about our restorations, this is a really great example. This is in French Creek. These are our spawning pools. Adults can come in, spawn there. We can have baby fish living in these. And we'll put a net here, 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 and catch them leaving into the main French Creek channel. Our reference are unmanipulated habitats. Remember, those are our natural habitats. You can actually see, if you look closely, the typhus starting to block that natural channel in that photo right there, which is pretty interesting. These are, again, all facilitated by our lovely collaborators and wouldn't be possible without them. So let's just finish off here with some data comparing the two types of habitat. So we have our spawning pool, which, remember, we created. U.S. Fish and Wildlife, TIBS, collaborations, we created these pools. Our references are green color, natural. So we can see that the pools are actually a little bit warmer. And this is a good thing. We have fish growth, prey items that thrive in this warm water. So we see a difference, likely related to some of the sediment and that gets exposed when we dig out these pools and heat up with the sunlight. And also, they're still relatively comparable. And right, that's a good thing. Our restorations are comparable to a natural channel that's already in the wetland. This is giving evidence of some success of our restoration. What about oxygen? Oxygen is pretty comparable between the two sites. I failed to mention that this data is during the emigration period, so that critical period for those baby fish. So we can see that our blue spawning pools create a little bit more oxygen, and that's great, despite them being warmer, right? So remember, warmer water holds less oxygen. Our spawning pools are warmer and they're holding more oxygen. So this is a good thing. Again, we're still trying to figure out what's too warm and what changes could, climate change could be too warm, but regardless, warm, oxygenated habitats that we have modified and restored are really positive for northern pike and other fish communities. Finally, looking at our northern pike catch and our northern pike size. So if you look at our x-axis again, we have our spawning pools, our modified habitats, and our natural channels. We can see that they produce fish. Our y-axis here, the length of the fish, they produce fish about the same size. This is just from this past summer. Historically, these sites produce a little bit bigger fish. And when we're talking about baby fish, bigger is better. It means you can't get eaten by another fish or a bird or another animal in the wetlands. Bigger is better at this stage in life. So we can see both sites produce about the same size fish. Historically, spawning pools a little bit bigger. This is great to see. Our restorations are impacting growth in a positive sense. We can also see we catch a lot more fish out of the spawning pools. This could be related to more open water habitat. Remember, we have those little pools within the wetlands. We have more water for these fish to live in and to grow in. So based on our northern pike here, it's clear that some of these habitats really positively influence them in terms of growth and catch and environmental conditions. That's a little awesome. That's about the size they be. They're very wholesome and cute, but they get much bigger, obviously. So just to wrap up here, what did we think about? What was important when we're thinking about Thousand Islands wetland nursery habitat? Environmental conditions, right? So each wetland is different. We can't just monitor one wetland and say that's it. That's the entire region. That's what this means. We need to see how that wetland was formed. What type of wetland? Does it have river influences? Things like that. So monitoring within a wetland, so looking at interior, exterior, and then monitoring between wetlands is key. This will help us understand restoration success as we do more restorations and learn more about manipulations in these wetlands, and then thinking about climate change and that balance between water temperature and oxygen. The fish community, we have evidence of emigration processes and that species respond differently. So we see evidence of baby fish leaving these wetlands to go live in the main river. We see evidence of different species with different tolerances and different life histories responding differently. And then finally, thinking about our focal species, right? We're at historic lows, sadly, but some of our restorations and some of the environmental conditions we're creating in these wetlands are helping and are having a positive impact. I hope to tease out more differences so we can really understand what is driving something like a northern pike's survival. This is Cranberry Creek, just absolutely beautiful. Tough to paddle through, but very beautiful. And then finally, what's next? So continued work with this environmental and this catch data. We can use things like data modeling to predict what's going to happen. So we can predict maybe a 2 degree increase in water temperature, and it can tease out these complex patterns. So you saw lots of different data. We can put that all together, and a model can help us tease out a trend or a pattern. More lab work, so that's what I do in the winter. I live on campus, essentially, in Syracuse. I don't actually live on campus, but I'm spending a lot of time in the lab working with our otoliths, looking at biomarkers to try to understand the fish's past, assessing our stocking success, and then using lab experiments to control what we're seeing in the field in the lab setting to understand more details about what's going on with fish, oxygen, water temperature. And then finally, continued monitoring on the river, right? I'm lucky to come to a place that has all this long-term monitoring data, and we need to continue to do that. So we want to expand our data logging arrays to more than 4 wetlands. We can expand them, maybe some more embayments, so we can really understand what is happening to the wetlands in the Thousand Islands. And finally, more hopefully restorations based on all this data and understanding what is successful and what isn't. And lastly, I just want to talk about all my acknowledgments on my collaborators. I want to thank all our different collaborators that give us not only money but access to these sites and interest in getting these projects done. So the public, everybody here, you all contribute to this and you really help us get this stuff done. As a scientist, it's really, really amazing to see such an engaged public, an engaged group of people that we have in the Thousand Islands. It's not like this everywhere you go. So this makes our job easier. So I want to applaud people for that because it really helps us facilitate work and get amazing research and protect these ecosystems. And then obviously our TIBS staff. So Dr. John Farrell really giving me this opportunity as a graduate student and others to contribute to protecting the St. Lawrence River and the ecosystems here. And I can't forget all the SUNY ESF students, graduate students, undergrads who help out doing field work, collecting this data, and also local students. So we work with a lot of younger elementary school students for ichthyologists for a day where they learn all about fish on the river. We have some high school students come out and help us. So we're really trying to get everybody involved. Science isn't just for scientists, it's for everybody. So yeah, I would just like to finish up that, these wetlands need to continue to be monitored, protected, and successful, positive, healthy wetlands really shape the future of the fish community on the St. Lawrence River. So they're very important to continue to monitor and protect. And with that, I will take any questions. All right.
I have one question. Is there a diurnal variation in the migration from the wetlands?
That's a good question. That's actually something I think I'm going to be exploring next summer. So with these nets, they're checked once daily. So we only get a snapshot of one night, a whole night though. So next year I'm thinking about doing some interesting net sets where, you set it for a couple hours and then you come back maybe in the middle of the night, things like that, and get at what you're saying. But when we check them now, it's likely, those fish have moved into that net and have left the wetlands between about, noon the previous day when the net is reset to when it's checked the following morning. So like a 24-hour period. So hopefully I can answer that question with some more studies. So I think it'd be really interesting. That's a really great question. I think it'd be really interesting to, check the net throughout a diurnal period and see if it really ties tightly to that oxygen regime. So yeah.
When you build the spawning pools, about how long does it take before fish move into those?
That's a good question. So that's going to depend on a lot of different things, obviously. But that Eel Bay wetland, that was surveyed in 2021. I believe that project was completed in 2019, if I'm not mistaken. So that was, no more than 3-year difference, and we caught 16,000 fish from that wetland. So I think the old sort of quote, if you build it, they'll come. I think that really holds true here, especially in places that are really connected to the river. Fish find the habitat and use it. So that's great to see.
A follow-up question.
Sure.
Too. So once you've built the spawning pool, how long is it before the typhoid takes back over?
That's a good question too. I sort of learned that myself this year, trying to get into some of these sites. So it seems like, I was in restorations that were over 5 years old and I could still access the pools. But definitely compared to some of the aerial imagery, the typhoid does move in. So this is something, this gets at this idea of monitoring, right? We need to keep keeping an eye on where the typhoid is and where it's moving, because it can easily clog channels. So I guess it just depends, maybe if you have a nice current or a wind, it's going to push typhoid mats into a spot quicker than others. So yeah, it depends, but I've seen restorations last more than 5. Before my time, before I got here, there are restorations that are still functioning perfectly well. So.
I think I heard you say that you were monitoring the success of stocking rates.
Yep.
Does that include northern pike?
Yeah, so this past April, we stocked a bunch of marked northern pike, so they're otolith. We'll have a little highlighter mark on it. So we stocked known numbers into these habitats. So we stocked known numbers into the pools. We stocked known numbers into the regular natural channels. And then we came back 3 weeks later and put the nets in. And we caught between 4 wetlands this past spring, over 700 little baby pike. So I'm in the process, obviously we don't keep all of those, but I'm in the process of looking at a subsample to understand what % of our fish survive.
What would you consider a successful rate?
Well, so we've done this study once in the past, and in combination, it was just in French Creek, and they had around 5% survival if you added everything up. Yeah, well, but remember, these are little baby fish, so there's a lot of them at once, and a lot of them are going to die.
Roughly how many did you stock?
This past spring was 150,000.
And I think 5% of those.
Yeah, hopefully. So that was between French Creek, Point Vivian, Chippewa Creek, and Cranberry Creek.
Okay, well, I'll be waiting on my tip-ups this spring.
Yeah, luckily they won't be too big. They won't be too big yet, but yeah, it's important to figure out. And this winter, I'll get an assessment of like % survival, right? So yeah, that's stuff I'm still working through.
Some of the warm spawning pools had a higher oxygen content than would be expected. I take it from the temperature of the water. What enables or causes it?
Sure. So the big thing in those spawning pools and in the wetlands in general is the shallow water. So compared to the main river where you have, water depths below 10 feet, 5 feet, things like that, all these locations are really shallow water. So that water can heat up really quickly. In addition, in those spawning pools, we have a lot of exposed dark sediment. So this dark sediment can absorb that light, and it seems to do it a little bit better in the pools than in these little channels. So I think that both of those things kind of contribute to those spawning pools being pretty warm. And again, that's a snapshot of the long-term monitoring data we have on this, but it seems to be pretty positive for our spawning pools. Yeah.
Just curiosity, and maybe the time interval, maybe before you started doing your research, but a few years ago we had record high water. So we heard today about what, how that might affect the fish populations. Do you have any concrete numbers?
Well, so we're working on some stuff right now with those high water levels, and we did see differences in actually oxygen based on the high water. And in terms of catch, I think it kind of varied by wetland, but we do see both high and low catches in high water. I think it just sort of depends on where you are and the severity of it. There are some spots in French Creek that are so low, then as you get closer to the main river, it's a lot more water. So I think it sort of depends on where you are, but those high water years actually sort of potentially tended to promote more low oxygen, which was something we didn't expect. We're still working through some of that data now, but we think that high water flushes in a lot of that decomposing typha, and that decomposing typha to decompose takes up oxygen. So we're still working through details on water levels and the exact effect. But clearly there's an influence, and a physical influence too, right? If an adult pike physically can't get into the wetland or there's not enough water, there's not going to be baby fish. So yeah, I think it's sort of this interface of where the wetland is, the season too, right? I mean, gosh, in these high water, low water years, the climate is impacting that as well. So I think it kind of depends on where you are, but we've seen both high and low catches during these years. But definitely the high water makes it easier for fish to get into the wetlands compared to low water.
So I have a question.
Sure.
Your pools were warmer,
Mm-hmm.
which should indicate greater productivity and more food,
Mm-hmm.
but the size of the fish was very similar
Mm-hmm.
between the pools and the reference.
Mm-hmm.
What do you owe that to?
So there's two things. So first off is that data was just this year. I think if I put it in the historic context, we would see the larger fish coming from the warmer pools. The other thing that could be influencing that is just that the pools can hold more fish, right? So there's going to be more fish competing for the same resources. So that may indicate, that may have an effect in terms of, the overall group is about the same size, but there's more of them leaving the wetland, if that makes sense.
Mm-hmm.
So yeah, I think it's a combination of both. I think if you plotted it with more long-term data, you would see the spawning pool size, but then I also just think the spawning pools can hold more fish. So you have more fish, but they're a little bit smaller, but they're still very comparable to the fish coming out of the natural habitat.
And what caused the multiple peaks in the emigration?
So that's really tied. I'm diving into the data a little bit more, and there's some really interesting peaks of oxygen and water temperature during those times. So I'm thinking I have to do a little more analysis to figure that out, but that could be influencing it. And then it's just different species. So different species are going to have different egg incubation rates. The adult fish are going to come in at different times. They're going to grow at different rates. They're going to have different tolerances. So like in Flynn Bay, for example, I saw a huge emigration of really sensitive fish early. And then later in the year, you see peaks of other fish, like a brown bullhead, that wasn't leaving the wetlands at the beginning. So I think it just depends on what species it is, and that's why it's really important to note the species and not just take fish catch for fish catch. You need to understand what you're actually catching and how that relates to the conditions we're seeing.
Just upstream from the French Creek Bridge
Mm-hmm.
is a very large section of exposed bottom.
Mm-hmm.
There's very small channel.
Mm-hmm.
Is that going to affect the spawning in the spring?
It depends. So I've actually seen if you even move up further in French Creek, maybe this is where you're talking about, but I've seen a spot in French Creek where the typha, there's just a very tiny channel left. So yes, it could potentially. We're still trying to figure out, and I think we speculate that some of these fish can swim under the typha.
But in this case, it's mud flats.
Oh, it's mud flats.
It's
Yeah.
mud
So
flats.
that's right now we'll see what it's like in the spring when the water goes up, but definitely, if a fish can't swim through it, it's not going to be able to go in.
Right.
So yeah, it could definitely affect the spawning numbers. And we actually set nets to capture. I didn't cover this, but we set nets early in spring to capture the adult pike, just to get a sense. And some years we hardly catch any. Some years we catch 50 plus adults in one spot in French Creek. So it just sort of, I think, depends on the year. But yeah, if that stays mud flats, fish are not going to be able to move through that. So they need water.
Yes.
Anything else? I really appreciate everybody spending their Friday night learning about science, and yeah, thank you.
Thank you.